Hello, everyone. Welcome to the fourth session of the BGE Day at the Biodiversity Genomics Conference 2023. In this session, we're going to be talking about opportunities for increased utilization of genomic data for biodiversity characterization and conservation. We have for that invited researchers that are in the forefront of innovation and development for biodiversity genomics. And in these presentations, we're going to be catching technological innovations and logistical opportunities. The 5 talks are going to run and start with the DNA barcoding for long-term pollinator biomonitoring with Mara Lawniczak and Clare Carvell from the Wellcome Sanger Institute and the UK Centre for Ecology and Hydrology. Then we're going to go to the DARK-TAXON barcoding with Rudolf Meier from the Natural History Museum, Berlin. Finally, monitoring biodiversity from airborne DNA with Elisabeth Clare from York University. Martin Fischer from UKCEH, Zurich. After that is going to be talking about monitoring genetic diversity, where he's going to tell us about the pilot study in Switzerland using genomic data. And last but not least, we're going to have Kok van Oosterhout from the University of East Anglia talking about genomics for the conservation and recovery of threatened species. At the end of this, all the talks that are going to be shown pre-recorded, we're going to have a Q&A session, so I invite you to put your questions as early as possible in this Slido. See you on the other side.
Hello, it's a pleasure to be giving this talk jointly with Mara Lawniczak from the Wellcome Sanger Institute. I'm Clare Carvell from the UK Centre for Ecology and Hydrology, and I lead the UK Pollinator Monitoring Scheme, which is funded by JNCC, our Joint Nature Conservation Committee. I'm going to start off talking about UK POMS and then hand over to Mara, who's going to talk about the Bioscan UK project and a new collaboration between our 2 projects. So, in the UK and globally, really large-scale biodiversity monitoring has been restricted to quite a few taxa, for example, the birds, butterflies and moths. And these long-term data sets have been vital in identifying threats and declines in those taxa, but they still give us an incomplete picture of the environment. And many of our most highly diverse groups among the insects, for example, are understudied, despite, of course, playing such key roles in ecosystem functioning. And this is largely due to a lack of taxonomic capacity and the cost associated with training and building that taxonomic capacity to identify usually small and tricky specimens. In the UK, we have a fantastic resource in terms of being able to measure the distributional change of species from ad hoc biological records that are collected by amateur naturalists as part of these volunteer recording societies. So here we see some data for bees and hoverflies. But these distributional data don't tell us still about changes in abundance, which of course is so important for ecosystem functioning. And while we only have data for bees and hoverflies, we're not understanding about change in the hundreds of other species that are pollinating wild plants and crops other than bees and hoverflies. So the UK Pollinator Monitoring Scheme was set up to try and address some of these challenges. It's the world's first monitoring scheme to be generating systematic data on the annual abundance of bees and hoverflies and other flower visiting insects, and doing this at a national scale. UK POMS has established 2 new large-scale surveys, which started up in 2017 after a period of design and testing. And those are the flower insect timed counts, which is a 10-minute observational count, which is very much a citizen science project, and I won't say any more about that today. And then we have a systematic 1 kilometre square survey using pan traps, which I will be going on to talk about. UK POMS has a very strong objective of wanting to engage individuals and communities in taking action for pollinators, and we're helping to provide evidence to the UK's 4 national pollinator strategies. So the UK POMS 1 kilometre survey operates across a network of 95 1 kilometre squares. These have been stratified according to the 4 countries of the UK, and they occupy the sort of relative proportional representation of agricultural and semi-natural land across each of the 3 countries. So it's been designed to test broad UK-level changes in pollinator groups and in some species for which we can pick up enough data from the surveys we're undertaking. The sites are co-located with other national UK monitoring schemes, and works on the principle of volunteers adopting a square. So most of the squares you can see, particularly those in blue, are surveyed by volunteers who are trained by the POMS team. The ones in red are available for adoption, and they're covered by paid surveyors as part of the team. The protocol involves setting up 5 sets of pan trap stations across the 1 kilometre square. So in the map you can see on the top left, for example, and it's designed specifically as a kind of feasible 1-person, 1-day protocol that can be undertaken by 1 person in a day. Around each pan trap station, a number of other environmental variables are collected, such as floral abundance, and we collect weather information on the weather at the time of the day, and so on, that we can use in our models. And so this is John, one of our great volunteers at one of our sites, AIRBORNE site in Cambridgeshire. So in terms of what happens to these pan trap samples, getting the sort of pipeline for processing them has taken us a while to really kind of get working well, but we've now processed more than 335,000 insect samples. Those samples, consisting of 5 sample pots, 1 from each trap station per site, are sent free post by the volunteers, and they will come into the CEH labs at Wallingford in Oxfordshire, and they're then sorted and processed by our team on the CEH lab bench. We take out the bees and hoverflies, which are our target taxa, and so we're averaging around 18 bee and hoverfly specimens per survey, but there's a whole lot of other material, other specimens, other insects that are coming out of the traps, and we call that the bycatch. The bees and hoverflies are identified by a team which we've now expanded to around 7 or 8 taxonomists who are obviously have the expertise in identifying those bees and hoverflies to species, and we're very careful with the QA and checking of their IDs before we publish the data. But of course, there's now potential for us to look at using DNA barcoding methods to supplement the traditional taxonomy that's already taking place, and this is where the new collaboration comes in. So I mentioned about the whole series of other insects and invertebrates that we collect. This pie chart just shows you the scale of that difference. So the majority of a pan trap sample is in fact made up of the Diptera, the non-syrphid or the non-hoverfly flies, and various other insects. So really our target insects are only occupying a small amount of the sample. We've modelled the 5 years of the data so far using generalized linear mixed models, including some of the environmental variables. And you can see here, we're able to plot a general kind of trend or predicted count with a measure of uncertainty, which is the shaded line, for each of the different target groups. On the left, this is a plot of abundance for everything in the pan trap. And again, you can see here this grey line and the brown line representing all the other insects and the non-bees and flies that we're not yet able to identify as species. The plot on the right shows you changes in richness of bee and hoverfly species over that time series, and you can find out more in our annual report, which is available on the POMS website. So again, recognising that we had such a large archive of other insects, we were lucky enough to have a collaboration with Alfred Vogler and his team at the Natural History Museum to start looking at some barcoding of these samples. Initially, we undertook some individual Illumina barcoding of the bees and hoverflies within the pan traps to see how well they matched the taxonomic identifications from the conventional taxonomy, also following a previous paper by Thomas Creedy et al., where we looked at developing that workflow. And in this case, from 42 of our sites in a single year, there was a 95.2% match of the OTUs to the conventional taxonomists' identifications. We also, in this project with Alfred and his team, looked at a whole community COI-based meta-barcoding of all of that non-target bycatch, the non-bees and non-hoverflies that were coming from the pan traps that we had in our archive. And there was about 207 pan trap samples that were collected from, again, these 42 sites in a single year. What that showed, I mentioned we do morphologically identify to group level what those taxa are in the lab, just so we have an idea. That was where the initial pie chart was generated from. That indeed the composition of the OTUs that were generated by that meta-barcoding was very, very similar in composition to what we had identified from the lab, as you would expect. So there was a good correlation there, but we identified huge numbers of OTUs, mostly coming from the Diptera, from that initial exploration using the meta-barcoding approach. And for example, we can start to do some very preliminary analyses looking at whether landscape type is showing within those samples as structuring the communities, and a quick PCA on the sort of relative abundance of each of those species OTUs in the samples suggests that potentially, if you look here, the black representing agricultural sites, the red representing semi-natural sites, there is a signal that we have structuring of the communities that's showing through these genetic species-based identifications. So we may therefore have a really good tool for assessing insect community change at this national scale. But of course, in this project, these mixed samples were not allowing or enabling us to provide information about the individual abundance of species in those samples. And this is where I'm going to hand over to Mara to talk about our new collaboration. Thank you.
Thanks, Claire. Right, so we are in the midst of the beginning of a long-term project that we call BioScan. And this project is dependent on partnerships across the country, where our partners are operating malaise traps each month for about 24 hours. And in the catch bottle, there's just 50 mils of ethanol, and we just try to plate out. So our partners volunteer to plate out. You can see on the right a 96-well plate, each specimen that ends up in that 24-hour catch, so that we can assess with the diversity and the abundance of what has been caught in that 24-hour period each month. So they record key information, and then they ship their samples to us at Sanger. We then non-destructively extract the DNA. So we image each insect by zooming in on the well and taking a photo. The photos aren't great, but they give us some sense of what is in the well. And then we sort of leach the DNA out through a non-destructive approach, which means that we can keep the specimen for later confirmation or investigation. And we have about 100 microliters of DNA, of course, varying concentrations depending on the size of the insect. And we just use 0.1 microliters of that then to go forward with DNA barcoding using CO1. So that means we have a huge DNA biobank that remains for additional work on DNA barcoding of other barcodes and for genomes. Next slide, please, Claire. So right now we have 40 traps actively catching across the country. You can see our map to the left, and 20 more are about to launch. So our partners are onboarded and they're getting ready to start trapping. And we want to thank all of our partners, but especially the pilot partners that are listed in the bar below, who really helped us establish this process over the course of a couple of years before we launched into the full project. So you can see the POMS map to the right of our BioScan map. And so partnering with POMS is a super exciting addition for both of our projects, because it's got this potential to add a kind of a time series retrospectively. So they've been working for 5 or 6 years already on this project, and we can compare malaise and pan traps. On the right, you can see a drone picture taken by Bill Irwin from one of our partner sites in Shapwa Keith, just of the malaise trap, and kind of, there's a wide range of different ecologies that the traps are active in. So we're really excited to start looking at the data that is produced. Next slide, please. At the moment, we have, so this is real data. This is from the first sort of 10,000 specimens that we've DNA barcoded. On the right, you can see this sunburst, and the purple, it sort of goes out in taxonomy. So the center is the orders, and that big purple half of it is all Diptera, and then there's a lot of Hymenoptera and a variety of other orders represented. And then on the outermost ring, each place there's a line is a different bin or species identification. And this is just the first 10,000 specimens. So we have a huge wealth of information now that we can start to look at in space and time. And so from our pilot partners who were trapping at a smaller scale over the first 2 years of the project, you can see the bar chart at the bottom. And this is just a very high level view of species abundance that we can examine. And you can see the seasonality here, really. So there's all sorts of possibilities here in which we're working on an interactive website that is open for anybody to look at. And these are the sorts of features that we'll be building into it, where people can compare between sites or months or species, et cetera. And we are now actually actively sequencing 10,000 specimens a month. So while this is just the first kind of tranche of it, a lot of data is coming. Next slide, please. So one of the exciting things, as I mentioned, is that we still have a lot of DNA remaining. So we've got this really rich biobank that we can go back to for additional barcodes. One of the things that we're particularly excited about, for example, with the POMS study, is to actually go back to the samples and look to see what kind of plants we can detect using plant-based barcoding on those insect specimens. And even more exciting in some respects to me is the genome sequencing potential of this giant, what will turn into a 1 million, biobank of 1 million insect DNAs. While the DNA is typically very low quantity, there is sufficient length so that our non-destructive approach was really focused on trying to make sure we released long molecules as best as we can. And so in the lower left here, you can see a femtopulse profile of the DNA, and you can see it's very small, but actually that little peak kind of in the middle that sort of triangulates up is sitting at 40 kb. So our molecules tend to range between 10 and 40 kb. And although there's not a lot of DNA there because we've non-destructively leached it from the specimens, that length enables us to do amplification-based long-read sequencing. And so this is an example of a real bioscan specimen. This was its DNA profile. We sheared it. We put it through ultra-low input PacBio. This is its Kamer profile in the center, and you can see, it's looking like we're getting good coverage and even relatively good representation of the DNA in this specimen. And then Bioscan told us what the species was. It's a Chironomid midge, and the resulting reference genome is in 275 pieces with an N50 of 1.1 megabases, and it's a 125 megabase genome. So we still have a challenge in terms of using the biobank from Bioscan to create reference genomes, which is Hi-C is really important in creating reference genomes. And of course, we've now non-destructively extracted the specimen. We did actually go back and test whether we can grind up the specimen that remains after the DNA leaching to see if we can generate good Hi-C data, but it doesn't look very promising, which means that for these specimens, we're going to need to find another individual to put through Hi-C. Next slide, please, Claire. And that's it from us. Thank you very much to all of our POMS and Bioscan funders, partners, teams, landowners, and volunteers. And we'll be happy to have a discussion at the end of this session.
Thank you very much for the invitation. Today I want to talk about high throughput species discovery with robotics and ONT. And for this, it's important that we remind ourselves what animal biodiversity really looks like. In the following cartoon, we are looking at the biomass distribution in terrestrial habitats. What we see is that the dominant taxa in terrestrial habitats are arthropods and annelids, followed by livestock, so the things that we eat and drink, us, and everything else is in terms of biomass not terribly important. If we modify this cartoon to show species diversity, then we actually have to only enlarge two taxa. One is the arthropods and the other one nematodes. Yet the other taxa that are also on this diagram shrink to next to nothing. Is this reflected in our biodiversity data? And the answer is unfortunately no. If you look at the Global Biodiversity Information Facility, then most data is available for birds. Almost like 3/4 of the data that is available is bird data, while arthropods have next to no data. Why is there so little data for arthropods? Well, it has to do with the taxon biases that are deeply entrenched in biodiversity sciences. We tend to look and work on things that are ugly or that are pretty large, that are pretty diurnal, and pretty endangered. Although pretty endangered species in terms of ecosystem services are probably not terribly important, with some exceptions, of course. So we are delivering biased data for a problem that needs unbiased information, which is the upcoming or the existing biodiversity crisis. Non-biologists have actually figured this out. So here's like Forbes and the World Economic Forum writing a report on ugly species that also deserve biodiversity protections. And what do they show? They show an arthropod. So let's change that by actually tackling arthropods, and especially tackling those arthropods that we call DARK-TAXON because they are very rich in species and also very abundant. Samples look kind of like this. And most of the species in this jar will be species that are DARK-TAXON in the sense that the number of specimens is extremely high, the number of species is high, and most of the species are unidentifiable, and a lot of them not described, and yet they represent a very large amount of different functional diversity. Who are they exactly? So are we talking about all of arthropods? No, we are not. This is here a study based on Malay strap samples from all over the world. When we looked at the content of these Malay strap samples with mega barcoding, we found out that there's a convergence in dominance and neglect in flying insect diversity. The same 20 insect families dominate all samples, more than 50% of the specimens and 50% of the diversity. At the same time, they're also very largely neglected by biologists. Not even the taxonomy is worked out. What are the top 10 here? So here I'm showing you the top 10 taxa, and they're doing all kinds of different jobs, some of them more pleasant than others. So they're definitely important for ecosystem services, also important for human health. So here are two taxa that are known to be vectors. There are two taxa that are phytophagous and also transmit plant viruses, also to economically important plants. There are two taxa that are parasitoids, controlling insect populations. There's one taxon that is critical for keeping our freshwater environments functional. There's one taxon feeding on mushrooms, and there are two taxa that are doing a wide variety of different things. Clearly, these are important for ecosystem services, but they are dominant and neglected at the same time. So that's what we want to address in the Center for Integrative Biodiversity Discovery at the museum in Berlin. If you have a problem with large numbers of specimens and large numbers of species, you need something like a factory, and that's what we're trying to build here. This factory consists of three different parts. The first part is a robot. This robot helps us moving individual specimens, but it also takes images of individual specimens, and I'll show you the details in the next slide. If we take lots of images of specimens where we don't know what species they are, then we haven't really gained much. So we need to assign these images to species level units. And we do this with DNA barcoding. And here we use nanopore sequencers because we are in full control of when what is being sequenced, and we don't have to use service providers. Now, once we have enough images for a particular species, we can start training algorithms, AI algorithms for future identification just based on images without any kind of sequencing. So here's a robot at task. It has two cameras, an overview camera that identifies where the specimens are, a second camera that then takes individual images of specimens, then comes a pipette that picks up the specimen and moves it over into a microplate in preparation for sequencing. Here the same process again, overview camera. Here we see the different specimens. The diversity scanner right now is only working on specimens that are below 5 millimeter in size, but those are the most abundant and the most species-rich taxa in those kind of samples. So this is what generates lots of images automatically. Now we need to get the barcodes. This is a very trivial process now, and we have written a paper where we show how to do this at scale and very easily with very little equipment. That's why we call this really for everyone, by everyone, for everyone, because we do need to accelerate biodiversity discovery. You can sequence 10,000 specimens on one flow cell. We of course have to manage the specimens too, because we want to be able to do follow-up work on the vouchers. And in order to manage the specimens, we are using biobanking equipment, like this kind of compound, but you could use any other equipment. This particular machine here, or this particular cabinet, can house 200,000 vials, so we could put 200,000 vials specimens in there. The way it works is that each of the vials will have an individual specimen. The vials are being sucked up. There is a QR code at the bottom of the vial. The QR code will be read by a scanner that is part of the compound. And then the vial is being stored in this gigantic carousel. That part is kind of trivial. What is important is that we can retrieve the samples, and we can retrieve the samples based on the QR codes that we give to the compound. And of course, we will retrieve them now by only exporting those vouchers that have identical barcodes. So we can start with an unsorted sample, then we do imaging, and then we do barcoding. And what we export are vials with specimens that are sorted to species level. And that gives us vouchers. And this is excellent for doing genomics. It's also excellent for getting trade data. I'll just give you a quick example here. This is now still from Singapore. So we did a study on mosquitoes there. Once you have the mosquito vouchers, you can sequence them further, and then you can find out what a particular species is feeding on. And this particular Aedes species is feeding on mammals, it's feeding on birds, and it's also even feeding on a frog. It's a kind of species that we have to be worried about because it can potentially transfer viruses from one clade to another. There are other species like this long-legged species where we don't have to be worried because this one is a specialist of mudskippers. So you see how important it is to have vouchers because we can do all kinds of follow-up work. Let me go to the last point, which is like training the AI algorithms. This happens the moment we have like enough images for a particular taxon. In this particular case, we trained the algorithms only at the family level because we had enough images for these different families. And the diversity scanner is able to identify all these taxa with about 90% accuracy without doing any kind of sequencing. The common taxa always will benefit first when we do this kind of AI training. Eventually, the AI training will go from family to genus to species. And as a matter of fact, we are now training the AI algorithms for the most common species found in an urban environment like Berlin. So ultimately, the species discovery factory will simplify further and will turn into a species biomonitoring factory where we can skip the DNA barcoding step. That's excellent news because that gives us more vouchers, but also better vouchers because they haven't even been touched with any kind of genetic technique. So how do we actually get started? Isn't it hopeless? When we look at a sample like that, we always think, well, may as well not even try because there's too many specimens and too many species in there. But it isn't really all that hopeless. So we have developed a 5-step program. We call it dark taxonomy. Which is kind of like a phase 1 of taxonomic exploration for a taxon that needs a lot of taxonomic exploration. Step 1 is we're getting fresh material from traps that are also used for biomonitoring. Fresh material because we can process it much more easily. And traps that are used for biomonitoring is an important point because we do want to resolve those species first that are also of importance for biomonitoring. So we may as well do taxonomy with the material that is coming from biomonitoring traps. The second step is we pre-sort the specimens with DNA barcodes, very much like what I already described. The third step is we are looking at the motos that we're getting with the DNA barcodes, and we fix those motos where the barcodes are not reflecting species boundaries. So we do integrative taxonomy with a second data source. Then once we have delimited the species, we identify or describe them. Here's an example from Singapore. We found 120 species of fungus gnats in a Malay strap, in about 100 Malay strap samples. The 120 species belong to 5 described species and 150 new species. So that tells you how badly known dark taxa really are. The vast majority of species are unknown. And we then went ahead and described the new species. Now it's important to figure out whether we have actually covered all the species that are important for biomonitoring. And that we do with a second test sample. So we are not having only the taxonomic sample that we used for doing the revision. We also then take a second test sample coming from the same Malay straps, but different periods of collecting. And then we compare whether we have the same species in the test sample that was also in the taxonomic sample. So the whole thing is very successful if the test sample mostly consists of those species that we described based on the taxonomic sample. So what is the situation looking like? So here's a test sample, and indeed 79% of the species that we have in the test sample were already described based on the taxonomic sample. And only 21 species, 20% of the 21% of the species are only found in the test sample, and that constitutes, and they represent 20 species. More importantly, we have to look at specimens because biomonitoring is also about abundance. So what about specimens? Well, the success is even much more resound. 97% of all the specimens in the test sample belong to species that were described based on the taxonomic sample. And that's simply based on the fact that there's abundant species that are very likely to be in the taxonomic sample and the test sample. And then there's only rare species that are showing up only in a few samples. So only 3% of the samples belong to the 20 new species that we found in the test sample. They're all rare species. So we can actually go from unknown to ready for biomonitoring in 5 steps, and at the same time make a valuable contribution to taxonomy, because by doing these 5 steps and doing this first exploration with dark taxonomy, we lesser the taxonomic burden impediments, but we lesser them for a particular purpose, which is biomonitoring. Let me come to my conclusions. Society expects us to monitor biodiversity holistically. We're not doing that at all right now. A major challenge is getting unbiased data. We can get unbiased samples, but now we also have to evaluate these samples properly. And I think with decentralized sequencing, automation, and machine learning, we can actually take on the challenge of dark taxa. It'll be a major challenge, but it is a challenge that is doable as I showed you with our 5-step dark taxonomy protocol. And this is what we do at the Center for Integrative Biodiversity Discovery at the Museum for Naturkunde in Berlin. Thank you for your attention.
Thanks very much for the opportunity to present at Biodiversity Genomics 2023. My name is Elizabeth Clare, and I'm from York University in Canada. And I'd like to talk today about the opportunity of monitoring biodiversity from airborne eDNA sampling. In my lab, we work primarily with indirect sources of DNA, and over the last few years, that has mostly been in the form of environmental or eDNA, the form of DNA shed by all organisms into their environment. eDNA is most widely known from water, where thousands of studies have been published and a number of commercial industries have developed around the applications of aquatic eDNA. And some governments now include aquatic eDNA sampling in some of their analysis and regulatory structures. But eDNA was actually first recognized on land, and an explosion of studies have now found eDNA in a wide array of locations, from soils and honey to rain, blood meals, and left behind in footprints. And most recently, we have found it on leaves. You can wash the surface of a leaf to collect DNA, carried on the backs of insects, where they seem to have DNA from other animals and plants in the area that they actually carry around with them, and even in some strangely inorganic sources like rock and some forms of coal appear to harbor environmental DNA from their environment. The problem on land is not finding it, but figuring out the best source. And the best known cases are what we would call indirect sources. For example, we have tracked mammals by eDNA in the blood meals of things like terrestrial leeches, or by swabbing dung beetles because they interact with mammal feces. These are indirect, and we have problems with degradation and inhibition because the material has been digested. An analogous approach to aquatic eDNA sampling would be to target eDNA in the air as a way of surveying for local terrestrial life. However, until recently, this was primarily a theoretical field, with a few studies that looked at things like the detection of bacteria and pathogens, and a very small number that looked at plants and some fungi, mostly through passive collection methods like the ones in the pictures depicted here. We became interested in air in my group about 3 years ago, and our first goal was to show that under laboratory conditions, you could actually actively extract DNA from an air sample. And we chose to work with naked mole rats in a colony to give us the best possible chance of getting it work the first time. And the reason is that here you've got a group of individuals that are living in close quarters in a small space. And we had the idea that if it was ever going to accumulate in the air, that would be the condition you'd be able to detect it the easiest. And so we started there, sampling inside the burrows of naked mole rats and inside the room. And we were astonishingly successful at finding the DNA signatures in those air samples. So proof of concept that this was actually possible. The next step then is to demonstrate you can do it under more natural conditions. And the problem was coming up with a controlled scenario that actually replicates a real environment. What we chose to use was a zoological park, and this is Hamerton Park in rural UK. The reason we chose a park like this is that the animals are not native. So we're not going to mix them up with signals from the environment. If I was detecting a cow in rural England, I would have no way of knowing the source. But if I detect a tiger, there's only really one place it could come from. And so we walked around this zoo and we took air samples both inside their sheltered enclosures and then outside in their habitat. And we were astonishingly successful again at detecting the animals, both the animals inside their enclosure, but also the DNA of the animals moving through the park. Up to about 300 meters distance, we were still picking some of them up as the DNA floated through the air. So this gives us an idea that this can work at a certain range. We also detected some of the wildlife from the environment actually coming into the zoo. It's an open-end area they can walk through, and we found hedgehogs and bats and ducks and squirrels and the things that we would expect to find there. We've also tried some passive sampling. We teamed up with a group in Texas that use these remarkable little passive samplers that always turn into the wind. They're really, really clever. They accumulate dust, And they had looked at the dust for plant material. We looked at it for mammal material and bird material, and we found a relatively good correspondence with what we knew about the local ecology. And so these were taken every 2 weeks over an entire year, and we saw patterns in our detections that matched known activity patterns on the landscape and showed rare things as being rare and common things as being common. So largely matching what we knew about the environment, which is really good news for this being a good temporal signal. We then developed a new form of handheld monitoring system that we take out into the actual environment to do some really applied science. These are one of our models. We have a number of different models we use now. Some of them are more contained inside boxes and some of them are more open, but these run off batteries. And so you can carry quite a lot of them in your backpack and you can take them out and deploy them. And some of them will run for 2 days on one battery charge, which is really nice. We use these in controlled communities where we control who's there in different abundances. We've also used them in truly applied research. So this is a survey we've conducted of bat roosts in Central America. And in both cases, we were extremely good at recovering the biodiversity that we knew or expected to be present. So in controlled environments, we can look at the exact match between detections and presence. And in these applied surveys, we have past data on who lives in these caves to get an idea of what we're detecting is reasonable, and it generally is. And so from this, we can sort of say that we know that animal and plant DNA is being carried in the air. It can be passively or actively collected. It's reflecting recent activity on the landscape, and it's accurately describing complex communities, at least when they are contained. However, it doesn't really get to the problem in biodiversity science, which is this. This is the first field site that I ever worked on in my PhD in Costa Rica, and I don't think we're any better now than we were back then, and it was quite a few years ago now, at actually measuring biodiversity that's in this place. We don't have techniques that scale. And the problem for this is that really we don't have standardized methods. Most of our methods are taxon specific. And because of that, we don't really have standard metrics. We have basic metrics that we try to apply. And a lot of these studies are done very locally. And so we tend to extrapolate our data to try and understand larger patterns based on pilot data or small scale work. And the reason we sort of work on this operation is that we don't have a globally distributed infrastructure for doing this. And this is what a lot of our transnational agreements, like the Convention on Biological Diversity, ask us to do. But the challenge is we don't have the infrastructure for it. But maybe we were wrong. Last January, Joanne Littlefair and I were contacted by a physicist, James Allerton, and a metrologist, Andrew Brown, who worked for the National Physical Laboratory of the UK. They had seen a report on the zoo experiment that we did, and they asked us to explain it to them. They then asked if we had considered looking at air quality monitoring networks as a source of material. This is a picture of one of these devices that they operate on behalf of the Environment Agency in the UK, and these filter air at a standardized rate every week over and over and over again as part of large networks that monitor air quality. In this case, they're looking for heavy metals, arsenic, lead, things that are of risk to human health and are regulated in our air for human health surveillance. On the top in this image here is a new filter before it goes into the machine, and at the bottom is what they look like when they come out. Here we've cut a little quarter off of them, but they've accumulated particulates from the air. And so we set up an experiment with the National Physical Laboratory to see whether or not these stations are accidentally collecting eDNA as a byproduct of their regular operation. This is one of their network sites in the Scottish Heathlands, and they sent us 8 filters that had been collected and analyzed for the heavy metals, and we're now ready for disposal, basically. We also set up an actual controlled experiment outside Bushy Park in London, where this is not a regulated station, so they could manipulate the amount of time it sampled for. So we looked at an hour and a day and a week of accumulation, the week matching what they do under the normal system. These were astonishingly rich in environmental DNA. In just those 2 locations and 17 samples, we detected 180 different taxonomic groups represented as DNA captured on the filter, 80 genera of plants and fungi, 34 species of bird, and 26 different mammals, all of which were known to exist in the local environment. I don't know of any other method in biodiversity science that can simultaneously assess fungi and plants and mammals and insects in the same way at the same time. Remember, we don't have generalizable methods in biodiversity science. And if you remember, we did this time experiment at Bushy Park, and that's the first sort of 3 columns of this graph. And it makes total sense with how the behavior of these different taxonomic groups would operate. So with plants, after a day, you've pretty much captured them all. You don't get any more if you sample for a week because they're all physically present. There's nothing new coming and going. But with things like the vertebrates, which are in yellow here and hidden underneath, so there's the green accumulating for an hour and then a day, underneath that we have the vertebrates, and they continue to accumulate because more animals and birds and mammals are coming and going from that environment over the course of a week, and so you detect more things the longer you sample for. What's also really interesting is that out in the Scottish samples here, just look at the plants, for instance. There's a week of sampling at Bushy Park versus a week of sampling at the Scottish heathlands. And they're pretty amazing because the diversity is about the same. And what we didn't know at the time was that those Scottish samples had sat for upwards of 8 months without preservation. And so what this is suggesting to us is this DNA is robust. It continues to hang around for weeks or months, even when you don't actively preserve it with DNA in mind. The vertebrate diversity is lower, but we would actually predict that because this is a less diverse area than Bushy Park, which is basically an urban forest. And so this is a really good indicator that the regular operation of these networks is going to be compatible with the preservation of DNA. Now we have new questions. How far does the material travel? What is the temporal persistence of these signals, and how well do they match known distributions? Well, it turns out the United Kingdom is probably the perfect place in the world to test this, because we discovered they have upwards of 150 sites that are taking absolutely identical weekly samples, which means that on an annual basis, the UK is capturing nearly 8,000 air quality samples, which we now believe are harboring environmental DNA. That's an astonishing richness of potential biodiversity records. And to try and map that, the UK is also uniquely good at knowing about the distribution and abundance of various major taxonomic groups. They have plant atlases and surveys of large mammals, which will tell you to the square kilometer the abundance and distribution of these species. We can use that to try and map out the DNA signals and look at how well they matched. But this is not a UK phenomenon. This is a map of the air quality monitoring stations regulated by Airbase under EU law. Remember, these are transnational agreements on air quality monitoring and particulate matter. And so we have absolutely replicated systems in dozens of countries that are taking the same types of measurements. This is also not a European thing. Here's a map of the air quality monitoring stations the EPA regulates. This is just the ones that measure at the same size fraction and seem to save the samples in the same way as the British ones. They are operating in exactly the same way, taking measurements every few days. I now have maps like this from all over the world. They are in the public sector, they're in the private sector, they're hidden in some of the most amazing places where biodiversity is rich. And mostly they're operated by chemists and physicists and metrologists. What we may have here is a globally distributed, semi-automated system collecting samples of terrestrial eDNA every day, everywhere, all the time. This is the global infrastructure collecting standardized samples that we've been looking for. Of course, they're not perfect. No method in biodiversity science is perfect, but this is the first thing I have ever seen which could scale to measuring biodiversity at the level of countries and continents. And they will be collected whether we analyze them or not, because they were not collected for biologists. They were collected for air quality assessments. These networks have been quietly sampling like this for half a century or more, and in some countries, they have saved all of them, which means we have the tantalizing possibility of studying biodiversity and the dynamics of populations through time. Thank you very much.
So I would like to thank the organizer for giving me the opportunity to present our pilot study for a monitoring of genetic diversity in Switzerland using genomic tools. But why is genetic diversity that important? So genetic diversity is the fundamental level of biodiversity. Biodiversity has 3 levels. So it is the genetic diversity on which builds up species diversity and ecosystem diversity. So therefore, genetic diversity is the raw material for evolution, and it allows species and population to adapt and persist in a changing environment. And therefore, it's crucial for long-term survival of all the populations. Genetic diversity contributes to stability of ecosystem, as it has been shown in eelgrass meadows here. And eelgrass meadows which have higher genetic diversity also have higher species diversity living within these eelgrass meadows. Or it also maintains ecosystem services, as shown in bees. So bees which have had higher genetic diversity had a better immune competence and therefore could do better their job to pollinate all the orchards or all the vegetables. So therefore, genetic diversity is a key element of biodiversity and should therefore be conserved and monitored. However, there is not a lot of knowledge available on how we can monitor genetic diversity. That's why we wrote 2019 a feasibility study for Switzerland, how we could monitor genetic diversity. We also did a stakeholder analysis to see what are the needs of the stakeholders. This paper was published in 2021 in Conservation Genetics. And we discussed that we should maybe monitor 50 species across different time horizons in Switzerland. But starting with 50 species would be a big burden. So that's why I decided to do a pilot study where we start monitoring 5 species in Switzerland. So we decided to monitor species which occur in habitat of national importance, like the hairstyle cottongrass, which lives in peat bogs, or the Carthusian pink, which lives in dry meadows, or like amphibians, like the Lawniczak toad, or organisms which live in anthropogenically modified landscape like the yellowhammer, or also butterflies, which are amazingly, or all insects which are endangered for extinctions. So this whole project is financed by the Federal Office for the Environment in Switzerland. And we had to design a new sampling strategy, the best way to sample genetic data in the landscape. We came up that the proportional stratified random sampling strategy is ideal. What do we mean with proportional? So we proportionally, given the occurrence of the species, sample them proportionally to the 6 biogeographic regions in Switzerland. And as you can see, we were quite successful. So we sampled more than 158 population across Switzerland and more than 1,200 individuals. And as you can see, the sampling is really more or less random across Switzerland for the butterfly in orange, yellow for the yellowhammer, brown for the toad, yellow for the Carthusian pink, and in green for Eriophorum vaginatum. Additionally, we de novo assembled and annotated all the 5 genomes. So we did a de novo assembly using PacBio, HiFi data, and OmniC data for some of the species. And for most of this assembly, we nearly reached chromosome level assembly quality, which is really crucial for further analysis because it increased the accuracy of the estimators we are looking for, but it also reduces computation time on your clusters. So the genomes we assembled ranging from 446 megabases up to 3.8 gigabases for the toad. And as you can see, for most of them, we nearly reached chromosome arm level or chromosome level assemblies. And the BUSCO values are also, I would say, quite good with up to 98.8%. All the 1,200 individuals have been whole genome resequenced using Illumina technology, and we aimed for a coverage of about 14x. And with this data at hand, we were able to estimate genetic diversity for all the 5 species in Switzerland. So what you see here are the bar plots of genetic diversity for the 5 species, just summarize them. And as you can see, the butterfly has the highest genetic diversity and the toad has the lowest genetic diversity. However, I think it's very important that these results should not be interpreted without the context and knowledge of species biology. So it does not mean that the butterfly is completely healthy and the toad is completely bad in state. So we then defined 5 genetic indicators to estimate such changes of genetic diversity over time. So one is, of course, genetic diversity. We are looking for waters and theta, or site-specific waters and theta, then genetic connectivity, inbreeding, where you're looking for runs of homozygosity, the adaptive genetic diversity of these populations, as well as, of course, the effective population size of each population, because that's where drift acts on. So we are currently calculating all these values. As you can see, there are still some NAs, so it's still work in progress. But for all the 5 indicators and all the 5 species, already get the first values for all this averages for species, for population, but even values for each individual. Another attempt we are using is trying to go back in time with samples from collections and museums and try to get back in time up to 200 years. And so we have collected 200 butterflies from collections and 200 cotton grasses and have processed them in a clean lab and to get as much DNA as possible and get good genetic data. And also those have been sequenced for whole genome with a whole genome resequencing approach. Working with such old DNA up to 200 years in age, you need to work in clean lab, or at least it's better to work in clean lab. And we have developed a new protocol for butterflies where we can work with a single leg from a collection to get enough DNA. Just as a short glimpse, so for these 200 individuals we tried for the Areophilum vaginatum, we were able to successfully sequence 157 individuals, which are all the triangles here on the map. And we can compare them now with 310 contemporary Areophilum vaginatum samples, which have been sampled in as populations as shown in the dots. So if we look now overall already, because this is still work in progress and we're still analyzing them, but overall you already can see that genetic diversity from roughly 1900 has decreased until 2021. What I found even more fascinating is that contemporary genetic data also gives you information about genetic diversity in the past. So if you look at this map here, as greener the dots are, as higher is genetic diversity, in this case for the cotton grass, and as redder, as lower genetic diversity, meaning, okay, we see here is a bit hotspot of genetic diversity in Switzerland for these cotton grass, and here maybe it's a bit lower genetic diversity, which could also be linked with evolutionary lineages. And so it would mean that, okay, we have very good genetic diversity here in this area, which of course is not completely true. So if we now use GO-NE to estimate effective population size, recent effective population size back in time, so we can go and estimate effective population as back about 100 generations in time, you see that also this population slowly is decreasing in the effective population size. For some other population, it's even more severe. So we had a stable population and then a massive collapse about 40 generations ago, and then stable low population sizes. In some areas, we see that genetic diversity was never as high as in these areas, but also there we had a collapse about 50 generations ago. So I think that is amazing, but such estimates you only can infer if you have nearly chromosome level assemblies and whole genome resequencing data. So, as mentioned, we can see a decrease of small or effective population size in this grass species. Another nice example how genomic data can be used is the yellowhammer. So, as you remember, overall genetic diversity of yellowhammers in Switzerland is quite high, but still we are able to detect local inbreeding in these birds. And so what you can see on this map now are in green increasing breeding pairs and in orange are decreasing breeding pairs, and each dot here represents the inbreeding level of some individuals in these populations. And if you look a bit carefully, you may identify that it's especially in areas where the number of breeding pairs in the last 20 years have decreased, we see some sort of an increase of inbreeding. Maybe better shown here where we have on the y-axis the estimated change of breeding pairs in the last 20 years per square kilometer, and on the x-axis, the inbreeding coefficient FRO. And you can see areas where we have a decrease of breeding pairs, we see an increase in inbreeding, and these values get as high as 14% of the genome being homozygous, which is a quite high value already. And even more fascinating with these genomic tools is that we can estimate the most recent common ancestor of these inbred individuals. And for these inbred individuals, we can date that this inbreeding has happened more recent than 25 generations ago, which nicely fits with the estimates here from the breeding pairs changes over time. So overall, this pilot study gave a lot of insights. So first of all, monitoring genetic diversity using genomic tool is feasible, and an ideal sampling strategy is such a random proportional sampling strategy because it gives you really changes over the whole Switzerland and gives you a good impression on how genetic diversity has changed or will change in the future. And we showed that on a technical point of view, it is possible to do such a monitoring using whole genome sequencing data. And we now get really a contemporary baseline of genetic diversity for 5 species. And using herbarium and collection samples, we were able to show that it's possible to go back in time and see changes of genetic diversity over time. So what is the benefit of such a monitoring of genetic diversity? I would say it is really an efficient early warning system for changes. So we were able to show that we have increased inbreeding, like in the yellowhammer, or recent inbreeding. We were able to show decrease or small effective population sizes in the grass. And what I have not shown, we also can show that we have lost connectivity between some of the populations. So this helps to implement conservation measures like plan or promote target connectivity measures between populations which have lost their connectivity. The designation of regions with diversity of protection, so areas where we have a high genetic diversity should be protected. And something also I've not shown is that we also can infer these evolution lineages being present in Switzerland. And so these evolution lineages should be carefully looked at for artificial greening and reintroduction projects. So with this, I'm at the end of my presentation. This work has been supported by the Federal Office for the Environment, as well as ETH Zurich and the Foundation Geobotanical Research Institute Rübel. And we got technical support from the Genetic Diversity Center Zurich, as well as the Functional Genomics Center Zurich. And if you're curious about more information, have a look at our webpage, gendiv.ethz.ch, write me an email or have a look at Twitter, what now is called X. So many thanks for your interest, and I'm looking forward for your questions. Thanks.
My name is Kok van Oosterhout, and I'm professor of evolutionary genetics at the University of East Anglia in the UK. Today we'll be talking about genomics for conservation and recovery of threatened species. So genomic erosion is a threat that is experienced by many threatened species, particularly those that are experiencing population size decline. It's not just a loss of genetic diversity, but it's also an increase in the expression of the genetic load, basically bad mutations that are recessive in the genome and that become homozygous because of small population size and inbreeding and genetic drift. In addition, there's maladaptation, basically due to changing environmental conditions or, for example, domestication of zoo animals to zoo conditions. And there's the risk of genetic introgression that can happen after hybridization, when two species become in secondary contact. And this can happen, for example, due to climate change or range shifts or human-mediated transport. Now, all of these effects are basically of genomic erosion are not directly assessed by the red list of the IUCN. And for that reason, it may be possible that, or it's likely that the red list is underestimating the extinction risk of many threatened species. So at the most fundamental level, to understand what is really causing genomic erosion, we can consider the effects of habitat change and environmental change on the evolutionary forces. So there are 5 evolutionary forces that shape all the genetic variation. 3 forces that generate new variation, that is mutation, recombination, and gene flow, basically that make new variation in the genome. Then there's selection that basically shapes this variation, and then there's genetic drift. Genetic drift is due to, for example, small population sizes or bottlenecks or founder events. And particularly, genetic drift is a very severe threat in many populations because habitat destruction and causes population size decline, and that causes more genetic drift and inbreeding, and that has a severe impact on genomic erosion. Now, over here we have a simulation of the island of Mauritius, and these red dots represent pink pigeons, and also the yellow dots. So what we see here is that the forest coverage from 1773 to today, and what you can see is that due to habitat destruction, there is increasingly less habitat available to the pink pigeons, causing population size decline. Now that's also shown here in these graphs. For example, on the right in panel A, we see the forest cover in square kilometers over time. And what you can also see is that the census population size declines very much in line with what actually happens to the amount of forest cover. However, if you now look at the population genetic statistics in the genome that you can observe in the genome, then you see that actually the inbreeding coefficient only starts to increase much later after the population has already collapsed quite significantly. Similarly, the heterozygosity is also responding much later. So there's a real drift debt and a lack in the response of these genetic parameters to the population size decline. Now, this is a paper we've published, Pinto, Alessandro Pinto, has written the computer simulations, which was published recently in Conservation Genetics. Now, if you now look also at the genetic load, what you can see is that actually the genetic load responds even later than most neutral variation. And this is largely due to the fact that recessive mutations, deleterious mutations, tend to be present at a very low frequency. So in order for them to increase to appreciable frequency and to really change this, these kind of aspects of the genetic load, it takes a long period of genetic drift. So the drift depth for the genetic load tends to be even longer than that of neutral variation. Now, these are individual simulations that really try to understand how this affects the population viability of the pink pigeon. Now, if you look at the IUCN red list, what you can see is that actually the pink pigeon is doing pretty well. It's assessed as being vulnerable, which is the second highest endangered category. So critically endangered, endangered, and then vulnerable. So actually, it's not as bad, although the green status assessment says it's critically depleted, basically because it has had such a severe bottleneck. So currently the population is quite stable. It's about 500 or so, just over 500 individuals, but the species recovery score, according to the green list, is only 17%. So it still has a long way to go to actually recover. Now, when you look at the actual census population size, and that is of course what the red list really categorizes, then you can see that the number of males and females has increased rapidly. Here from the late 1980s, when there was a severe bottleneck, from about 12 or so individuals surviving in the wild, to say around about 500 on total males and females presently. So of course, this has resulted in the downlisting of the species in the red list. However, if you now look at the genetic effects or the genomic effects, then you can actually see that this demographic decline really has a long-term impact. And we published this in this paper in Conservation Biology last year. Now, what it basically shows is that over the actual recovery period that the population increased in population size, we can actually see that the observed heterozygosity declined. So even though the numbers increased, heterozygosity declined. And this is clear evidence of strong genetic drift. Now, what you can also see in the genome is that actually you have large runs of homozygosity, and this is particularly damaging if in those runs of homozygosity there are some recessive deleterious mutations, because those mutations become then expressed as homozygotes, and that can reduce the fitness of the individual. So this is really what causes inbreeding depression. Now, we have published a few papers to discuss this in the literature. This is basically a perspectives paper, and this is a review paper you might find interesting. It really talks about genomic estimates of and how you can estimate the genetic load using techniques that have been developed for humans and model animals in conservation in threatened species. And this is a paper that's also perhaps interesting that talks about the purging and the accumulation of genetic load in conservation. Now, to really start to work with the genetic load, we can actually calculate the presence of deleterious mutations in ultra-conserved elements. And this is work done by a PhD student of mine here, Sam Speak. He is a bioinformatics student and into genomics, and he has quantified the genetic load, trying to assess how the genetic load works out when you start to cross individuals. So basically, what is shown here are the different chromosomes. And this is a zoomed in version of chromosome 1. And all the black lines are ultra conserved elements. So basically areas in the genome that are very slowly evolving, that are shared between very diverged animals. So mutations in those areas are probably severely deleterious for the individual, because that part of the DNA, the genetic code, has been so well conserved. So any mutation is probably bad. So what he has now done is he has, so this is basically a zoomed in version of one of the ultra conserved elements. And over here you can see the CAT score, basically the mutation impact score for those positions. And this is actually the ultra conserved element. Over here there's a gene next to it. So you see that you have high scores in the ultra conserved element. Now, what he has done, Sam, is to try to identify these mutations, calculate the total genetic load, and try to minimize this by making particular crosses that would have a lower load expressed in their offspring. Now, this is how it looks like in a very kind of a figurative style. So these are individuals 1 to 6, and they are crossed with other individuals, including with themselves. That's, well, in this example possible. And what you can see in the squares that are red, these are combinations that have a high genetic load in their offspring. So if you cross individual 5 and 6, well, then you get a high genetic load. Or if you cross 5 and 4, you get a very high genetic load. But if you cross individual 6 with individual 2, well, then you have a very low genetic load, or realized load, basically the realized load of mutations that become expressed. So blue are good breeding pair combinations, and red are the worst breeding pair combinations. Now, by using this algorithm, you can now start to make the best crosses and reduce inbreeding depression and maximize fitness of the offspring. So we can also identify now the best individuals for release in the wild. So from zoos, to identify which individuals are actually best suited to minimize the amount of inbreeding depression in the wild. Now, we have done this also in computer simulations first to examine different breeding regimes, the minimized kinship regime, the minimized load regime, and minimizing both. So minimizing kinship basically is minimizing the relatedness according to pedigree records. The minimized load regime is basically picking 2 individuals that are have the lowest genetic load and breeding those. And then the combined regime is trying to optimize both. Now, in red, the bad mutations, in blue, the neutral mutations. Now, what you, of course, want to have is that you want to have low bad mutations, high neutral mutations, because that is more genetic variation and more adaptive evolutionary potential. Now, in the minimized kinship regime, you indeed have a lot of neutral variation, but you also have a high genetic load. So that is not ideal. The minimized load regime, yeah, you can reduce the amount of bad mutations, here red, but it also reduces the amount of neutral variation. Now, this combined regime where you optimize both, that actually manages to get the best of both worlds, a low genetic load and high genetic variation. Now, so what does this really mean for conservation? Now, if you can imagine that you have the genetic load here of a population on one axis and inbreeding on the other axis, then you have the different colors which indicate the extinction risk, with green low extinction risk and red high extinction risk. Now, suggest your population starts somewhere here, so it's not very much inbred, but it has a high genetic load because it was a large population with a lot of variation, also a lot of bad mutations. And now genetic drift would basically push this population to more inbreeding. And actually, of course, there's some purging, there's some reduction of genetic load because of this inbreeding and also genetic drift. Now, conservation genetics has typically just pushed back at this, trying to minimize the amount of inbreeding. Now, with conservation genomics, actually, you can also start to select against this genetic load. And this would not only reduce the amount of inbreeding, but also the genetic load in the population, making the populations more resilient to future genetic drift. Now, we've also done this kind of experiment in silico using computer simulations, and this is work by Hernan Morales. So this is computer simulations in SLIM. We're testing the different effects of genetic rescue of the pink pigeon populations with zoo birds, experimenting with 3 different regimes. Genetic rescue, so really introducing individuals with the right genetic makeup. We have also demographic rescue and the no rescue regime. Now, very clearly, the genetic rescue regime, where we apply this approach that we minimize inbreeding and genetic load, is the best, causing a lower genetic load, theoretically at least, in the population. Okay, so in summary, genomic erosion poses a risk to the long-term viability of many threatened species, and it's not just the loss of genetic variation, but also it affects genetic load, maladaptation, and hybridization. Now, this genetic load can be estimated for individuals and for breeding crosses, so that you basically predict the fitness of the future offspring, and you can hopefully reduce inbreeding depression in those. And it can also be used for the management of captive population, in particular to identify the best individuals for genetic rescue. Well, thank you very much for your attention, and also thanks to all these people, particularly Sam Speak and Hernan Morales and the several funding bodies. Thank you very much.
Hello, everyone. Welcome back. I hope you have enjoyed these 5 amazing talks. We have all the speakers here with us. We're going to start a Q&A session for the next 20 minutes. So I would like to first thank each one of you. These are amazing presentations, and the developments, I mean, they're really game-changing. And I think we're going to develop a little bit more here with the questions. So I will start with Mara and Claire. Ed Ricketts is saying, lovely talk. And how do you think you might extend your methodology to sampling of marine taxa? He wants to give an example of the Sea of Cortez.
Thank you for the question, Ed. I think it sort of depends on what the motivation there is. If it's to understand spatiotemporal change, I mean, I think first we would want to have a great understanding of the specimens, the species that are present, using single specimen-based approaches like Rudolf is talking about. Maybe Rudolf wants to answer this question too. And then I think it would be excellent to implement some of those kind of high throughput approaches that Rudolf was mentioning as well for marine species. Rudolf, do you want to say anything to add to that?
Yeah, I think one should really start doing much more single specimen based barcoding and less meta barcoding, because I think this gives us the vouchers that we need in order to continue with the exploration. Because it's not about knowing what species there, but also what it does. And I think for that we need vouchers. So in general, I think there has been not enough emphasis on doing specimen-based work.
Okay, and you think that the diversity that is found of invertebrates in the marine environment would allow for similar procedures as you guys are doing with arthropods?
I mean, it depends a little bit on what kind of invertebrates we're talking, because some of them are difficult to image, some of them are very difficult to extract without destroying them. So I was recently talking to a colleague who works on nematodes, and it's much harder to get the imaging and the sequencing done. But Mark would know much more about that. So I think there are some special challenges to different groups. I'm taking the easier challenge, which is the arthropod challenge. It's a huge challenge in terms of species numbers, but in terms of having a nice exoskeleton, it's actually making my life a whole lot easier.
Great. So Rudolf, just sticking with you here, Mark is asking, saying brilliant, first of all, and what do you estimate the global requirement for your biodiversity factory to be? How many installations and how much would each cost to install and run at scale?
Yeah, so I think what is really still missing is a major frugal science push. I mean, the fact that we're still using plastic consumables for this, I think, is stupid. I also think that PCR machines that are being used right now are not suitable for a full-blown rollout. The original way of doing PCR was water baths, which is a much better technique, much more frugal technique for doing this at a large scale in different environments. I also think that it wouldn't be a global rollout the way we would do it in developed countries, but it would be much more targeted towards particular environments. So in many places, it's not even clear what are the crop arthropods, what are which areas are highest priority, where do we need to know the diversity first. And the rollout, from my point of view, should be very specifically to questions that are also being phrased by whoever needs to use the techniques or wants to use the technique. So it's like a two-pronged thing. One is like making it a lot cheaper, because I think what we're doing right now is completely too complicated for what is needed. I think doing PCR and sequencing is really very simple, and we are using overly complicated tools. And the second part is to target specific questions, because then it doesn't get overwhelming. If I want to do a global rollout of this and cover all of the world, I think this is not something that can be done. On the instrument development side, we do have a problem with getting funding to even like get something like our diversity scanner robot to be commercially available. There's still some lack of funding for getting certain devices out that would be quite useful for doing biodiversity monitoring. So that's one of the challenges that we're facing right now.
Thanks, Rudolf.
How much does it cost? Like 10K will get you a barcoding lab, but I think at 3K should be able to give it to you. So then anybody can do it.
Great. So now we go to Beth, who is trying to simplify things. Beth, here Mara is asking, have you looked at the arthropod diversity in the air monitoring samples, and what DNA extraction approach are you using on the filters?
Yes, Mark had also asked a question about that. We didn't have wonderful recovery on our first attempt at that, but that was mostly a PCR failure as opposed to, I think, lack of DNA. We've had much better success at amplification in data we're now waiting to get back from the sequencers. We are using a system where we soak the filters in phosphate buffered saline overnight before we then just extract from the material as if it's a tissue sample in a QIAGEN kit. It seems to be working, and we had developed that for hand samplers, and it seems to have translated well into the filters. We've tried some other systems with isopropyl ethanol to precipitate DNA. It didn't increase the yield at all. So just a basic extraction seems to work. In terms of arthropod diversity itself, even with sort of not well amplified samples the first attempt, I think we had 8 orders of arthropods represented, possibly more than that. And the more recent amplifications have been stronger. So I think that the DNA is there. I think we just didn't really recover much of it the first try.
Thanks. And now going to Martin. Petra Korlevich is asking for the historic specimens. Did you take into account ancient DNA damage or low coverage that might be skewing the heterozygosity quantifications?
Yes, of course, we took all of this into consideration, and working with historic DNA is really a minefield. So first of all, we also sequenced all the samples 6 times more than contemporary samples. Then we did all the damage correction. We did test a different approach with hard calls, with low coverage approaches. If you do it properly, most of these or all of those approaches gave very similar results, which is already good, at least to see that these results are stable. But you need to take a lot of things into consideration. You need to specifically develop SNP panels that allows to have calls from both samples, contemporary and historic, because the DNA is much shorter. They don't map as good as contemporary data because the reads are only 40 to 70 base pair in size. They are only single-ended. So there's a lot you need to take into consideration. Also the mapping, because the reads are shorter, you need to correct for reference biases and so on. So there are tons of things you need to consider. And we tested a lot of, not yet all, so we're still analyzing data, but we took all the roads you can try to with hard calls, with low coverage approaches, with angst and so on. So yes, we did a lot of different testing. Luckily, whether it's hard calls or low coverage, we always get very similar results. So that's at least a good thing, but it's a minefield. There's a lot of testing needed and a lot of adjustments. So yes, we did.
Thank you. Carl, I have a question of my own. There are several around the world, in all sorts of countries. There are reintroduction and translocation of species that are endangered or for which the populations have shrunk. I think in many cases they are trying to do a demographic rescue, as you have showed us. In some cases it might be a genetic rescue. Very rarely this will involve any genomics at all. So if you were to start this for these populations that are now, the programs that are now translocating, how would you use genomics in the short term efficiently to guide this translocations or reintroductions?
Thanks, Camila, for that question. I think it's a really important question to answer because, yeah, we are right now at the point that we can start to do this. So there's a few issues that you can have a look at. First of all, genetic supplementation becomes very efficient when you can start to supplement the population with genetic variation that has been lost in the wild. So one of the things that you would need to do then is do a resequencing analysis of both the wild population that you want to supplement and your captive population that you want to reintroduce from, to identify whether there are genetic variants that have been lost in the past that may be still present in the captive population, and that you can then use to supplement the native population, the wild population with. So that's particularly useful when you can also use museum samples. So what Martin was saying about ancient DNA, museum samples are very useful as a baseline to identify what was the population really before it started to decline. And then the second thing, which I think is an important aspect, is identifying mutations that might actually be fixed, deleterious mutations that may have been fixed in your wild population that you may are able to mask by reintroducing individuals that are not fixed for those bad mutations, but they have still the wild type allele present at those genetic loci, because that will reduce the amount of inbreeding depression in the wild. And similarly, of course, or conversely, you have also a risk of introducing bad mutations that are fixed maybe in your captive population that you don't want to introduce. So that is another thing that you want to avoid. And again, with genomics informed conservation and the sequencing of those samples, you may be able to identify that. And then lastly, you can of course identify whether the population you have in captivity is indeed the same population from which you want to supplement. So is it indeed the same ESU, the same evolutionary significant unit or conservation unit? So just to double check that indeed you are reintroducing the animals or plants at the right place and the right environment.
Great, thanks a lot. So now connecting barcoding and genomes, there is a question for Mara from Rob Waterhouse. What parameters would be the main ones to consider for deciding which species to seek a second specimen for high-C data generation? And do you see, like maybe it's a top-up of my own, do you see this as an impediment in some level to use the type of sampling that is done for barcoding for reference genome sequencing?
Well, we're trying to make the extractions as high throughput as possible, so we don't want to separate specimens into different components. We're depending on a lot of volunteers across the country to plate these specimens out for us. So effectively asking them to do something extra is a lot. So we could ask them to separate each specimen with the anticipation that we'll get DNA from part of it and we'll have part of the tissue remaining that we can use high-C for. But I think our project is so much at the early stages right now of trying to figure out what are the boundaries of what we can do with the samples, the way that we are currently handling them. So to answer Rob's question, I guess the test that we did selected just 20 arthropods from a variety of different plates and different partners. And these were actually part of a different project. A PhD student in my group was looking for microsporidia. So he was testing the extracts to look for presence of microsporidia. And so the project was really to try to amplify microsporidia genomes from these samples. But of course, we had to sequence arthropod too to get at the microsporidia. And so what we saw as the boundaries of this right now, it does depend on amplification. So it was using the PacBio ultra-low input approach. And it looks to be like relatively, genomes have to be kind of small, so about 200 to 250 megabases, in order to get good amplification coverage across the genome and good assemblies out. I think the high C part is tricky because, especially with the approaches where you're having to do some kind of barcoding first to know what the specimen is. But I mean, I see a really positive future with approaches like Rudolf's, where we can actually just use the images first and then have high quality specimens preserved in vouchers, and we can go specifically to the ones that we want to do targeted high C or long read sequencing on those.
Yeah, thanks.
Yeah.
So that's a nice connection to the question to Rudolf from Pete. Great talk, thank you. So what do you see as the minimal information requirements for the new species descriptions? Can you talk through your thoughts about what future species descriptions should look like?
I think the main thing is it has to have more images. It does have to have molecular data, and the text should be much shorter. That is like the short summary in a way. Because only 10% of all insect species are being described as DNA barcodes right now. So it's a very, very small number, and that's a shockingly small number for given the fact that a lot of insect monitoring will use DNA-based tools. So that's what I think is the most important thing. It's the most important thing that we are like making sure that the images and the molecular barcodes will be there. And then I think we should be thinking much more about common species and work much more on common species as opposed to like always on rare species, because that allows us to use AI for identification at a much earlier stage and then doing more genomics on it.
Okay, great. Beth, we have here also a question from Pete, which is something that I also thought about. So how to handle wind direction and then speed for interpretation? Also, I guess different ecosystems will have different levels of humidity and so on that need to be taken into consideration to how this is captured from the filters. So how much of an issue is this?
We don't know. Our best data so far suggests that the size range we're sampling in does not move very far. And so I don't think the wind direction speed is a big worry yet. But the really interesting thing about this is because we're basically relying on a system developed for this particle size, they have been deployed with really well-known particulate matter models that the physicists use because they have to sample this way already for pollutants. So we can basically rely on what they know about the very specific travel distance of these particle sizes to interpret what we're looking at. But the whole idea right now is to try and validate against the most amazing maps that Britain has of things to truth that. So we suspect because they were developed with this knowledge specifically about where to place them to maximize collection in a useful way for background pollution, that we're going to discover that the system is doing the same thing for DNA. Everything so far looks local, which is really good, but not a meter, probably on the order of hundreds of meters, maybe a kilometer. That's anecdotally what we've seen, but we haven't got the data yet to truth it. That's what the next sequencing run is about, is sampling on much wider scales and over longer time periods to figure out whether or not these particulate matter models the physicists have are the same thing for DNA-based systems. But since they're sampling the same size range, I suspect that's the answer.
Okay, thanks a lot. So, and Martin, both Rob and Chiara are super happy to see the pilot in Switzerland to fructify. So Rob wants to know to what extent the metadata that you're collecting for all your samples, geoclimate, etc. And can you use such metadata for integrative analysis that combine genetic genomic trends with abiotic factors?
So of course, we have some metadata related to these samples, which vary between the species. So for the birds, we have even for each individual, not only birds, we have individual coordinates. Then for the birds, we even have weight and sex and everything. And then for the climate data, we will work with GELSA data, and we will do some environmental association analysis to figure out adaptive genetic variation related to climate change. So this is still all work in progress. First, just inferring adaptive genetic diversity using RDA or LFMM analysis. And we also develop even new approaches, which allows maybe to even calculate adaptive potential of future climate change. So that's still all work in progress. And we really try to incorporate all this information, yes.
Yeah, thanks. So we have 2 minutes. So Kok, there is a question here for both you and Martin from Mark Platze, but I will direct to you first. So he wants to know about the genomic diversity decay lag that you talked about. And this, identifying the conservation targets. Okay, how does it interact with the landscape surveys through time in Switzerland, in your opinion?
So very, very briefly, so the conservation genetic summary statistics and the features that are responding to kind of genomic erosion, they differ depending on what kind of statistic you look at. For example, the number of segregating sites and an allelic variation that declines very rapidly. But if you look at, for example, gene diversity, heterozygosity, pi, that goes much slower. The genetic load goes even slower than that. So depending on which feature you look at, the rate of the drift death and the genomic erosion differs depending on what feature you look at in the genome.
Okay, thank you very much. And I would like to thank you all. I think there are many other questions here, so please try to answer them, see who made them. And thank you all for watching. I hope that you have learned as much as I did in this session. Yeah.